Hello viewers, welcome to my channel. Today we will discuss about the biochemistry question paper of GATE Excel 2022. Some brief notes regarding the question and the related options are also provided here, which will be beneficial for you. So stay tuned in this video. The first question, which of the immunocells listed below are agranular? P is eosinophil, Q mast cells, R monocytes, S T cells. The options are option A is P and Q only, that is eosinophil and mast cells only. Option B, Q and R only, that is mast cells and monocytes only. Option C, R and S only, that is monocytes and T cells. And option D, S and P only, that is T cells and eosinophil. Here the images of the four immunocells are shown. From these images we can see that the first two immunocells, that is eosinophil and mast cells, contain granules in the cytoplasm, whereas the last two, that is monocytes and T cell, does not contain granules in the cytoplasm. So the correct options will be option C, that is R and S only, that is monocytes and T cells. These two cells are agranular. Neutrophil, eosinophil, basophils and mast cells are granular, whereas the monocytes and lymphocytes are agranular. Next question. Which of the following enzymes is located in the outer mitochondrial membrane? Option A, citrate synthase. Option B, fumarase. Option C, monoamine oxidase. And option D is succinate dehydrogenase. The correct answer will be monoamine oxidase. Now let's discuss about these four enzymes. First one is monoamine oxidase. Oxidase. From this name you can easily understand that this enzyme helps in the oxidation reaction. This monoamine oxidase catalyzes the oxidation of monoamine, employing oxygen to cleave off their amine group. These enzymes are found in the outer mitochondrial membrane in most of the cell types of the body. The citrate synthase, fumarase, succinate dehydrogenase, these three enzymes are important in TCA cycle. Citrate synthase is localized in the eukaryotic cells of mitochondrial matrix. It is encoded by nuclear DNA rather than the mitochondrial DNA. It is synthesized using cytoplasmic ribosome and then transported into the mitochondrial matrix. Next one is the fumarase. It is found both the cytoplasm and the mitochondria of all eukaryotes. The mitochondrial isoenzyme is involved in the Krebs cycles, that is TCA cycles, whereas the cytosolic isoenzyme is involved in the metabolism of amino acids and fumarate. And the third one is the succinate dehydrogenase. This is very important because all other enzymes of TCA cycles are found in the mitochondrial matrix, whereas succinate dehydrogenase is present in the inner mitochondrial membrane. This enzyme is found in the inner mitochondrial membrane because it has an immensely important role as catalyst in both TCA cycle and in the electron transport chain. Note that the succinate dehydrogenase is the complex 2 in the electron transport chain. In eukaryotes, this enzyme is found in the inner mitochondrial membrane, but in prokaryotes, it is found in the plasma membrane. Next question. Which one of the following compound is not a translation inhibitor? Option A is chloramphenicol. Option B is cyclohexamide. Option C, puromycin. And option D is rifampicin. Here these four options are antibiotic. And among this antibiotic, the rifampicin does not help in inhibit the translation. Now let's discuss about this four antibiotic. Rifampicin is thought to inhibit bacterial DNA dependent RNA polymerase, whereas the cyclohexamide, puromycin, chloramphenicol all are translation inhibitor. Cyclohexamide is the translation inhibitor of eukaryotes, whereas chloramphenicol is the translation inhibitor in prokaryotes. And puromycin is the translation inhibitor in both prokaryotes and eukaryotes. Cyclohexamide exerts its effects by interfering with the translocation step in protein synthesis. The translocation is the movement of two RNA molecules and mRNA in relation to the ribosome. Whereas puromycin is naturally occurring aminoglycosidic antibiotic that inhibit the protein synthesis by blocking further extension of nascent chain and resulting in premature termination of translation. And the chloramphenicol inhibits microbial protein synthesis by binding to the 50S subunit and inhibiting the action of peptidyl transferase, thus preventing peptide bond formation. This mechanism also prevents the binding of amino-acyl tRNA to the peptidyl transferase active site. Next question. Which one of the following statement about the DNA polymerase III of E. coli is not correct? Here 4 options are mentioned and you have to choose that which one is not correct about the DNA polymerase III of E. coli. Option A is it catalyzes nicked translation. Option B, its absence is lethal to E. coli. Option C, it synthesizes a complementary DNA strand using a single-stranded template. And option D, it possesses 3' to 5' exonuclease activity. And the correct answer will be it catalyzes the nicked translation. That is, DNA polymerase III of E. coli does not help in the catalysis of nicked translation. Whereas its absence is lethal to E. coli because it is the primary holoenzyme involved in the replication activity. So if it is absent, it must be lethal to the E. coli. It synthesizes a complementary DNA strand using a single-stranded template. It is also correct because DNA is synthesized in the 5' to 3' direction using a single-stranded template and produce a complementary DNA strand. So option C is also correct. Option D is it possesses 3' to 5' exonuclease activity. It is also correct because the DNA polymerase III of E. coli possesses 3' to 5' exonuclease activity, that is the proofreading activity that corrects the replication mistakes. DNA polymerase III synthesizes base pair at a rate of around 1000 nucleotides per second. DNA Pol III is the component of replisome, which is located at the replication fork. Next, the enzymatic activities of DNA Pol II. DNA polymerase II possesses 5' to 3' DNA synthesis capability. It also has 3' to 5' exonuclease proofreading activity. DNA Pol II interacts with multiple binding partners common with DNA polymerase III in order to enhance its fidelity and processivity. Last, the enzymatic activities of DNA Pol I. DNA Pol has 5' to 3' DNA-dependent DNA polymerase activity that requires 5' primer site and a template strand. DNA polymerase I also has 3' to 5' exonuclease activity, that is the proofreading activity. So this proofreading activity is present in both polymerase I, polymerase II and polymerase III of E. coli. DNA polymerase I possesses the 5' to 3' exonuclease activity that mediates the nicked translation during DNA repair or primer removal. So the option which is incorrect for E. coli DNA polymerase III, that it catalyzes nicked translation because this activity is present in DNA polymerase I of E. coli. Another important draw of DNA polymerase I is that a 5' to 3' RNA dependent DNA polymerase activity. Next is about the RF value determination. A dye is allowed to undergo migration on a chromatographic paper using a solvent. The dye and the solvent front migrated 5 and 20 cm respectively from the point of origin. Now you have to determine the retardation factor, that is the RF value for the dye. So the RF value is distance travelled by the solute divided by the distance travelled by the solvent. Here distance travelled by the solute is 5 cm and distance travelled by the solvent is 20 cm. So the answer will be 0.25. That is the retention factor or RF value for the dye is 0.25. Now let's discuss about this RF value and its importance. Retention factor or RF value for a given substance should stay constant if the chromatograph is conducted under the same condition, that is the same mobile and same stationary phase. This enables the comparison of unknown material to known material. So by determining the RF value, one can identify an unknown substance with comparison to a known substance. They are the same compound if the retention factor for unknown substance differs from that of the known substance. RF value is always less than 1. An RF value of 1 or very close to 1, it means that the spot and the solvent front travel close together and are therefore unreliable. Retention factor value of a thin layer chromatography are affected by absorbent, solvent, chromatography plate itself, even the application technique and the temperature of the solvent and plate. And the more the polar compound, the more it will adhere to the adsorbent, and the smaller the distance it will travel from the baselines, that the lower its RF value. So for a more polar compound, the RF value is low. Next question is about the determination of value of isoelectric point. The pK value of a carboxylic and amino group of an amino acid with non-ionizable side chain is 2.17 and 9.13 respectively. The isoelectric point of this amino acid is, to determine this, the formula is isoelectric point for a particular amino acid is pK1 plus pK2 divided by 2. Here pK1 is 2.17 and pK2 is 9.13. The calculation is shown here, and by simplifying the value it will be 5.65. So the isoelectric point of this amino acid is 5.65. The isoelectric point is the pH at which a particular molecule carries no net electrical charge. The net charge of the molecule is affected by the pH of its surrounding environment and can become more positive or negative due to gain or loss of protons respectively. So if it can gain an electron, it become positive, or if it lost proton, it become more negative. Next question is about the molar absorption coefficient. The absorbance of a 5.5 into 10 to the power minus 4 molar solution of a tyrosine at 280 nanometer wavelength is 0.75. The path length of the cuvette is 1 centimeter. Now you have to determine the molar absorption coefficient at a given wavelength in mole inverse, semi-inverse. The absorbance A is equal to AM into BC, where AM is the molar absorption coefficient, B is the path length, and C is the concentration. Here absorbance A is equal to 0.75, path length is equal to 1 centimeter, and concentration C is equal to 5 into 10 to the power minus 4. By simplifying this equation, the answer will be 1500. So the molar absorption coefficient at a given wavelength is 1500 mole inverse, semi-inverse. Thank you. Please like, share and subscribe to this channel.